Trillions of microbes ready to invade our bodies lurk in the air, the water, the foods we eat, and on the objects we touch. Nevertheless, most of us are healthy most of the time and unaware of the constant battle between body and microbe. Even when infections occur, we almost always fend them off quickly. Our usual state of good health is due to three lines of defense: external barriers that keep microbes out of the body, non-specific internal defenses that combat all invading microbes, and our immune system, which assaults specific microbes. The body's first defense against microbes are the skin and the mucous membranes of the digestive and respiratory tracts. The dry, dead cells, filled with hard proteins that cover the outer surface of the skin, prevent most microbes landing on the skin from obtaining needed water and nutrients, while acids and natural antibiotics secreted by the sweat and sebaceous glands actively inhibit the growth of bacteria and fungi on the skin. The membranes of the digestive and respiratory tracts are protected by mucus, which contains antibacterial enzymes that destroy the cell walls of bacteria and physically traps microbes that enter the body through the nose or mouth. Cilia on the membranes sweep up the mucus, microbes and all, until it is either coughed up, sneezed, or swallowed. Swallowed microbes enter the stomach, where they encounter a combination of extreme acidity and protein-digesting enzymes that kills many microbes. Farther along the digestive tract, bacteria harmless to humans secrete substances deadly to other invading bacteria or fungi. Though these defenses are good, the warm, moist mucous membranes are more vulnerable to invasion than the skin. Disease organisms that enter the body through the skin or mucous membranes run into three nonspecific internal defenses: standing armies of phagocytic and natural killer cells, the inflammatory response, and fever. Monocytes and neutrophils are phagocytic white blood cells that travel through capillaries to wounds where bacteria have gained entry, then ooze out through narrow openings in the capillary walls. After leaving the capillaries, monocytes differentiate into macrophages, amoeba-like cells that engulf and ingest bacteria and other foreign particles. Neutrophils, the most numerous of white blood cells, like macrophages, also engulf foreign invaders such as bacteria and certain fungi. Both monocytes and neutrophils secrete molecules that attract other defensive cells to the invasion site. Natural killer cells are another class of white blood cells. They do not directly attack and engulf invading microbes, but rather identify and destroy body cells that have been invaded by viruses or that have become cancerous. Sensing molecular changes, natural killer cells bind to the compromised cell's cellular membrane and release proteins that pierce holes in it. Cytotoxic proteins then ensure the destruction of the abnormal cell by entering it and destroying its nucleus. Breaches of the skin or mucous membranes elicit an inflammatory response. Damaged cells release the chemical histamine into the wounded area. Histamine makes capillary walls leaky. Leaky capillaries cause fluid to seep from the capillaries into the tissues around the wound. The wound becomes red, swollen, and warm. Meanwhile, other chemicals released by injured cells initiate blood clotting that both blocks damaged blood vessels, preventing microbes from escaping into the bloodstream, and seals off the wound from the outside world, limiting the entry of more microbes. Still other chemicals, some released by wounded cells and others produced by microbial invaders, attract macrophages, neutrophils, and other phagocytic cells to the wound, where they engulf microbes, dirt, and tissue debris. The pus that collects around a wound consists largely of microbes, tissue debris, and living and dead white blood cells. If enough microbes survive the inflammatory response and infect larger areas of the body, a fever may be triggered. While severe fevers are dangerous, even fatal, average fevers of 38 or 39 degrees Celsius, 100 to 102 degrees Fahrenheit, are beneficial. In the brain, the hypothalamus contains temperature-sensing nerve cells that normally set the body's thermostat at about 37 degrees Celsius or 98.6 degrees Fahrenheit. However, certain white blood cells, in response to infection, release hormones called endogenous pyrogens that cause the hypothalamus to trigger behaviors that increase body temperature, shivering, increased fat metabolism, and a feeling of cold that causes a search for heat and warmth. The release of pyrogens also results in reduced iron concentrations in the blood, which, combined with the fever, slows the reproduction rate of many bacteria. Further, fever increases the activity of the phagocytic white blood cells that attack bacteria. Fever fights viral infections by increasing the production of the protein interferon, which is synthesized by certain body cells when they are invaded by viruses. Interferon released by these cells travels to other cells and increases their resistance to the viral attack. While often effective, these nonspecific defenses are not impregnable. When they fail to do the job, the body then mounts a highly specific immune response directed against the particular organism that has successfully invaded the body. More than 2,000 years ago, the Greek historian Thucydides recognized that occasionally someone contracts a disease, recovers, and never catches that particular disease again. The person becomes immune. With rare exceptions, however, immunity to one disease confers no protection against other diseases. Thus, the immune system attacks one type of microbe, overcomes it, and provides future protection against that microbe, but no others. This is why we refer to the immune response as a specific defense against invasion. The immune system consists of about 2 trillion specialized white blood cells called lymphocytes. Many are distributed throughout the body in the blood and lymph. Others are clustered in specific organs, particularly the thymus, lymph nodes, and spleen. The immune response results from the interactions among several types of lymphocytes and the molecules they produce. The immune response is provided chiefly by 2 types of lymphocytes, B cells and T cells. As with all white blood cells, B and T lymphocytes arise from cells in the bone marrow. Early in development, newly forming T cells migrate to the thymus, hence the name T cell, and differentiate. B cells differentiate in the bone marrow itself, hence the name B cell, and later migrate typically either to the spleen or lymph nodes. Immune responses produced by both B and T cells consist of 3 steps: recognizing invaders, launching an attack, and retaining a memory of the invader to ward off future infections. To understand how the immune system responds to invading microbes, 3 questions must be answered. How do immune cells recognize foreign molecules? How do they produce specific responses to so many different molecules? And how do they determine that a substance is foreign and not self, thereby avoiding destruction of their own body tissues? The key to the immune system's ability to attack invading microbes lies largely with proteins called antibodies and T cell receptors. Antibodies are either attached to the surfaces of B cells or dissolved in the blood plasma, where they are called immunoglobulins, often abbreviated Ig. T cell receptors are only found attached to the surfaces of T cells. Antibodies are Y-shaped molecules composed of 4 peptide chains, 1 large, heavy chain, and 1 small, light chain on each side of the Y. Both heavy and light chains consist of a constant region that is similar in all antibodies of the same class and a variable region that differs among antibodies. The combination of light and heavy chains results in an antibody with 2 functional parts, the arms, and the stem of the Y. The variable regions on the arms form highly specific binding sites for large molecules. Each binding site has a particular shape and electrical charge, so only a few, or perhaps only one type of molecule can fit in and bind to the site. In general, only large, complex molecules, such as proteins, polysaccharides, and glycoproteins, bind to antibodies. These molecules, or antigens, may either be attached to the surfaces of cells, the body's own or invading microbes, or dissolved in the blood or extracellular fluid, such as snake venom. As we will discuss shortly, the binding of antigen to antibody triggers the immune response. The stem of antibody molecules, which consists of part of the constant regions of the two heavy chains, determines the activity of an antibody. Though the constant regions in different antibodies are similar, they are not identical. For example, the heavy chain constant regions forming the stem of one type of antibody may attach the antibody to the plasma membrane of a cell. The constant regions of another type of antibody may bind to certain proteins in the blood, called complement, to promote the destruction of microbes. Antibodies perform two very important functions. First, antibodies are receptors. In an antibody's role as receptor, the stem attaches the antibody to the B cell's plasma membrane, while the two arms of the antibody protrude outward, sampling the blood and lymph for antigens. The binding of antigens to these receptor antibodies triggers responses in the lymphocytes bearing the antibodies. Second, antibodies are effectors. As we will explain shortly, certain descendants of B cells, called plasma cells, secrete antibodies into the bloodstream. There, the antibodies play several roles in neutralizing poisonous antigens or destroying microbes. Small differences in the stem, that is, the constant region, yield a number of true antibodies that serve different roles in the defense of the body. Some IgG antibodies attach to the membranes of invading microbes and form a coating on them that targets them for ingestion by macrophages, while other IgG antibodies bind to and neutralize toxins that enter the bloodstream. IgA antibodies are found in the heaviest concentration in the saliva and mucus secretions that coat the respiratory and digestive tracts and other areas of the body, while IgE antibodies largely bind to specialized cells embedded in the lining of the respiratory and digestive tracts, which release histamine during allergic responses. T-cell receptors are somewhat different from antibodies in structure and function. First, they are found only on the surfaces of T cells, not in the bloodstream. Second, they consist of two peptide chains of about equal size. However, the ends of the two chains protrude out from the T cell, forming highly specific binding sites for antigen, just as the ends of the arms of antibodies do. Third, T-cell receptors serve only a receptor function, recognizing antigen molecules and triggering a response in the T cell. They do not directly contribute to the destruction of invading microbes or toxic molecules. A human being may encounter thousands of different types of microbial invaders in a lifetime, from cold and flu viruses to the bacteria that cause strep throat and pneumonia, and the protozoans responsible for illnesses such as dysentery and malaria. The immune system recognizes and responds to this multiplicity of antigens by way of cells that produce millions of different antibodies and T-cell receptors, each capable of binding a different antigen. But how? Because antibodies and T-cell receptors are proteins, and proteins are encoded by genes, it would seem that a human being must have millions of antibody and T-cell receptor genes. But there are fewer than 35,000 genes in the entire human genome. How, then, do a fraction of these genes code for millions of antibodies and T-cell receptors? Two complementary mechanisms join forces to produce an enormous diversity of antibodies and T-cell receptors from a relative handful of genes. For simplicity, we will discuss only the generation of antibody diversity in B cells. Similar phenomena produce a large diversity of T-cell receptors. Genes encode parts of antibodies, not entire antibodies. These antibody part genes join together during immune cell development to form complete genes for the light and heavy chains of antibodies. The cells that give rise to B cells contain a few genes for the constant region of antibodies and several hundred genes for the variable regions. During the development of each B cell, the chromosomes containing the antibody part genes are cut, sections removed, and the remaining pieces joined together, bringing one variable region gene next to one constant region gene. Each cell comes to possess one constant plus variable light chain gene and one constant plus variable heavy chain gene. These constant plus variable genes are transcribed and translated into unique antibody molecules. Therefore, each B cell produces a single type of antibody, specified by the chance recombination of variable and constant region genes. Thus, the antibodies produced by any given B cell are different from most or all of those produced by other B cells, except, of course, their own daughter cells. However, after a number of generations, even daughter cells can be quite different from their ancestors, as the genes for certain antibody parts are prone to mutate rapidly and thereby constantly generate new antibody genes. The end result of gene recombination and mutation is that each B cell has its own particular antibody genes, different from those of most other B cells. At any given time, the body may contain perhaps 100 million different antibodies, so antigens almost always encounter antibodies that can bind to them. When an antigen enters the body, it may randomly bump into the variable regions of many antibodies, most of which will not bind to it. But with millions of different antibodies around, the antigen inevitably encounters one that binds securely to it. Antigen-antibody binding usually triggers changes in B cells that lead to the destruction of microbes bearing that antigen. But why don't the antibodies on at least some B cells bind to the body's own cells and destroy them? The surfaces of the body's own cells bear large proteins and polysaccharides, just as microbes do. Some of these proteins, collectively called the Major Histocompatibility Complex, or MHC for short, are unique to each individual, except identical twins. Because your MHC proteins are different from those of everyone else, they act as antigens in other people's bodies. This is why transplanted organs are rejected. So why doesn't your own immune system respond to these self-antigens and destroy your own cells? Apparently, it is the continuous presence of the body's self-antigens while the immune system cells mature. However, those immature immune cells that do produce antibodies or T-cell receptors that bind to the self molecules of the body are destroyed. Thus, the immune system distinguishes self from non-self by retaining only those immune cells that do not respond to the body's own molecules. If the body is invaded by microbes, the immune system mounts two types of attack. B cells provide humoral immunity through antibodies circulating in the bloodstream, while T cells produce cell-mediated immunity, as cytotoxic T cells destroy infected body cells. Each B cell bears, as we've seen, a specific antibody on its surface. When a microbial infection occurs, the antibodies borne by a few B cells will bind to antigens on the microbe. In reality, the process is considerably more complex than this, but this is the end result. Antigen-antibody binding causes these B cells to divide rapidly. The resulting cell population is genetically identical to the parent B cells, selected to multiply because they bind to invading antigens. The process is therefore called clonal selection. The daughter cells differentiate into two cell types, plasma cells and memory cells. Plasma cells churn out huge quantities of the relevant antibody, which they release into the bloodstream. Memory cells do not release antibodies, but rather, as their name implies, they remember antigens and therefore play an important role in future immune responses to a specific microbe. Antibodies circulating in the bloodstream can affect only molecules or microbes to which they are exposed. Therefore, humoral immunity defends primarily against bacteria, most of which never enter the body's cells, bacterial toxins, and some fungi and protists. Viruses can also be attacked, but only when they are outside of the body's cells, as during the first stage of an infection, or when they have finished replicating in a host cell, ruptured it, and released viral clones into body fluids. Thus, as we will see shortly, the primary defense against viral infections, aside from natural killer cells, is the cell-mediated immune response generated by T cells. Antibodies in the blood may affect antigenic molecules, microbes bearing antigens, and infected body cells in four ways: neutralization, promotion of phagocytosis, agglutination, and complement reactions. Neutralization occurs when antibodies combine with or cover up the active sites of a toxic antigen, such as a bacterial toxin, and thereby prevent the toxin from harming the body. Antibodies promote phagocytosis by attaching to and coating the surface of invading microbes. The protruding antibody stems help identify the microbes as targets for destruction by phagocytic white blood cells such as macrophages and neutrophils. Agglutination, or the clumping together of microbes, occurs as antibodies begin to bind to multiple microbes. As more and more antibodies link up with the antigens on different microbes, agglutinate results. This clustering of microbes enhances the speed at which they can be consumed via phagocytosis by macrophages and neutrophils. Complement reactions, which involve the numerous complement proteins found in blood plasma, are initiated by antibodies binding to antigens. Some complement reactions release chemicals that attract macrophages, neutrophils, and other phagocytic white blood cells to an invasion site and cause the invaders to stick to the phagocytic cells, enabling the phagocytes to engulf and ingest them rapidly.
Antibiotic-wise, they make lots of them. Do you know which one works the best?
Cell-mediated immunity is the body's primary defense when its own cells have become cancerous or infected by viruses. Cell-mediated immunity is also important in overcoming infection by fungi and protists. Several types of T cells contribute to cell-mediated immunity: helper T cells, cytotoxic T cells, and suppressor T cells. When their T cell receptors bind an antigen, helper T cells release hormone-like chemicals that stimulate cell division and differentiation in surrounding B cells and cytotoxic T cells. The chemical stimulation provided by helper T cells is critical to both humoral and cell-mediated immune responses, which explains why AIDS, which destroys helper T cells, is such a deadly disease. Receptors on the surfaces of the cytotoxic T cells produced as a result of stimulation by helper T cells bind to antigens on the surface of infected cells. The cytotoxic T cells then release proteins, often like those of the natural killer cells, that create large holes in the target cell's membranes. After an infection has ended, suppressor T cells shut off the immune response in both B and cytotoxic T cells. A small percentage of T cells formed during an immune response remain in the bloodstream as memory cells. Like memory B cells, memory T cells protect the body against future infection. Plasma cells and cytotoxic T cells do the immediate job of fighting disease organisms, but they usually live only a few days. B and T memory cells, on the other hand, may survive for many years. If foreign cells bearing the same antigens re-enter the body, they will be recognized by the appropriate memory cells. These memory cells will multiply rapidly, generate huge populations of plasma and cytotoxic T cells, and produce a second immune response. In the first encounter with a disease microbe, only a few B and T cells have the right antibodies or T cell receptors to respond initially. However, after the first immune response has run its course, the body retains hundreds or even thousands of memory cells capable of attacking that particular foreign invader should it invade again. This, along with the fact that memory cells can respond to antigens much more rapidly than earlier B and T cells, means that the immune response to subsequent infections is very rapid. In most instances, second or subsequent invasions by the same microbe are overcome so quickly that there are no noticeable symptoms of infection at all. Although cold and flu viruses appear to be exceptions to this rule, the immune system does in fact provide lasting protection against these viruses. The problem is that last year's flu virus isn't the same as this year's. As early as the year 1000, people in India, China, and Africa deliberately exposed themselves to mild cases of smallpox to acquire immunity to the disease. In 1798, Edward Jenner discovered that being infected with cowpox conferred immunity to smallpox. This discovery initiated the modern practice of immunization. In the late 1800s, Louis Pasteur extended the use of immunization to several other diseases by injecting weakened or dead microbes into healthy people. The weakened microbes do not cause disease, or at least not a severe case, but bear antigens that elicit vigorous immune responses. As in the case of real infections, the immune system produces an army of memory cells, conferring immunity against subsequent exposure to the living, dangerous microbes. These injections of weakened or killed microbes to confer immunity are called vaccinations. Today, many diseases, including polio, diphtheria, typhoid fever, and measles, can be controlled through vaccination. Smallpox, one of the deadliest diseases of all, has been completely eradicated from Earth, except for supplies held by Russia, the United States, and perhaps a few unknown countries, as a result of a vaccination program sponsored by the World Health Organization. Through genetic engineering, we now enjoy the prospect of manufacturing tailor-made vaccines. One method is to synthesize the antigenic proteins from disease-causing microbes. These antigens can then be used as vaccines without having to raise, isolate, and weaken the disease microbes themselves or to inject people with microbes at all. The vaccine for anthrax has been manufactured with this procedure. A second technique may be to insert the genes for the antigens of, for example, herpes, into the genome of harmless microbes, such as the cowpox virus. These designer microbes would produce herpes antigens without being able to cause herpes itself, and would thus be perfect for use in producing a herpes vaccine. Immune deficiency diseases result from the inability of the immune system to mount an effective immune response to infection. Probably the most common and most devastating immune deficiency disease is acquired immunodeficiency syndrome, or AIDS. Two viruses, named human immunodeficiency viruses 1 and 2, HIV-1 and HIV-2, cause AIDS by infecting and destroying helper T cells. Though AIDS patients produce HIV antibodies, for unknown reasons, they do little to prevent the progress of the infection. One hypothesis is that HIV spends so much of its life cycle inside cells that it is seldom exposed to the antibodies. AIDS does not directly kill its victims, but as the helper T cell population declines, the AIDS patient becomes increasingly susceptible to other diseases. HIV-1 and HIV-2 are retroviruses, viruses that have RNA, not DNA, as their genetic material. HIV consists of an outer envelope taken from an infected cell's plasma membrane as the virus leaves the cell, and two protein capsules. the innermost of which contains RNA and an enzyme called reverse transcriptase. The outer envelope binds to the plasma membrane of a helper T cell and allows the virus to invade the cell. Once inside, the reverse transcriptase copies the virus's RNA genome over into DNA. Because the normal direction of transcription in living cells is DNA to RNA, the virus's direction, RNA to DNA, is dubbed reverse transcription. The infected cell's own metabolic machinery then uses the viral DNA copy to make more viruses and more reverse transcriptase. This multiplication of HIV viruses eventually kills the infected helper T cell. The progress of AIDS can be slowed, but not stopped by drugs such as AZT or dideoxyribonucleic, DDL. These drugs are nucleotide mimics that fool the viral reverse transcriptase. When incorporated into a growing DNA chain, they stop further DNA synthesis. Ideally, the viral RNA is thus never copied completely over into DNA, and therefore a new virus cannot be synthesized. Unfortunately, these drugs are not completely successful in stopping reverse transcription. Further, they also interfere to some extent with normal DNA replication, and in some patients, they have very severe side effects. Because of these limitations and the huge scope of the AIDS problem, research into new AIDS therapies continues. A number of potential new therapies attempt to take advantage of the fact that HIV gains entry into helper T cells only after binding to a specific receptor on the T cell's plasma membrane. Thus, researchers are experimenting with flooding the body with compounds that bind to these receptors and prevent HIV from binding to them and gaining entry into helper T cells. Another approach involves producing an HIV vaccine, but this is a tricky business. As mentioned earlier, the antibodies normally produced by the body do not prevent AIDS. Therefore, an HIV vaccine would have to evoke a very different and more effective immune response than normal HIV infection does. Another complicating factor in the development of an HIV vaccine is that HIV has an incredible mutation rate, perhaps 1,000 times faster than the flu virus. Nucleotide sequencing of viruses isolated from AIDS patients shows that different people have remarkably different strains of HIV. Even more surprising, HIV can be very different at different times in the same patient. Nevertheless, trials of AIDS vaccines are underway in several countries, but unfortunately, none yet appear particularly promising.